Hi. In this video, we're going to see how glycogen is broken down. So in general, when we break down polysaccharides, sugar polymers, there are two ways you can do this. One is you can hydrolyze them, and the other one is to phosphorylize them. So one is hydrolysis, the other one is phosphorylases. They seem very similar, and they are, but they have different outcomes, and we'll see how that works out. So hydrolases, we've already seen a few of them. Lactase, for example, is one of them, right? That breaks down lactose in milk. So in that case, releasing glucose and the free glucose and free galactose. So hydrolases are very straightforward. Okay, so you have water attacking, R is the whole chain of something, of the sugar molecule, and you release just free sugar. There are two classes of hydrolases. The one that inverts the anomeric carbon configuration. So there's the enzyme. There is some general base catalyst for water to drive the whole to break down this bond essentially. So when you do that, however, so the water will attack from the side opposite, okay, of whatever the anomeric carbon is. In this case, you revert it. I mean, as shown here. Here the anomeric oxygen on the carbon was up, and here is down. Okay. How do you get the retaining? Well, you do two inversions. Two inversions equal the retention, and that's exactly what happens with this class of enzymes, the retaining ones. They just do two inversions. And you can play the same game. The enzyme directly, a carboxyl group in this case, participates in one of these reactions, and in the end, and then the water comes in, and then you have a retention of the configuration that you had when you started with. Again, thinking about these things is important. If you are to use the sugar in some way that some enzymatic reaction that depends on the anomeric configuration, then you may want to do a retaining mechanism. That's hydrolysis. What about phosphorylases? Well, who said that whatever is attacking here has to be water? Okay, what about free phosphate? Which has just a pretty good oxygen and it's going to attack it. And in this case, that's it. You break down the polymer, and what you generate is a phosphorylated sugar. Okay. Instead of a free sugar that we saw previously, let's say in this case here, okay, there is nothing on the one carbon other than the hydroxyl group, but here it's phosphorylated. So that's a key difference. And you can already see, I think you can guess where I'm going with this. So it is actually phosphorylase that breaks down glycogen. Okay. So when we break down glycogen, we don't hydrolyze, we phosphorylize. Not phosphorylate, phosphorylize. It's a different reaction. So there is no ATP here. It's just free phosphate, okay, shown here in green, which is coordinated properly by PLP, which we have seen before. There is another phosphate from PLP, and it gets coordinated. It helps to coordinate to put in the right place the green phosphates shown here in green that will actually attack the terminal glucose on a glycogen chain. Okay, so once this bond is broken, the phosphate will attack. You have a carbocation intermediate. Once the terminal 1,4 bond is broken, let's say on a glycogen chain, you have a carbocation intermediate at the 1 position, which really wants to be attacked by this phosphate. And what you end up with is this phosphorylated glucose molecule at the 1 position. Okay, and of course a cleaved glycogen chain which has 1 less glucose from what you started with. So phosphorylase, again, it's a PLP dependent enzyme, and the role of PLP here is not what we had seen in aminotransferases. It's really to help put the attacking phosphate at the right place. Why does it even proceed, this reaction? Because there is a ton of free phosphate in the cell. Okay, and so the mass action pushes it from left to right. And you also retain the configuration at carbon 1, and then we have seen how important that is. So because what you get is glucose 1-phosphate. If you want to use it in glycolysis, you have to move the phosphate to the 6th position. Otherwise, it's unusable. And so this glucose 1-phosphate is released phosphorylated, so you don't need to spend any ATP to activate, but you still have to work on it and put it on the 6th position. So a major reason for phosphorylasis is that, again, you don't need to spend ATP. Okay? So you spend ATP in the form of UDP when you activate the glucose to put it in glycogen, but when you actually mobilize this resource, you don't need to spend that extra ATP that you were spending in the case of bringing glucose from the outside. So that's pretty cool, actually. All right, so we'll see what happens to the glucose 1-phosphate in a minute. We've seen it already. I mean, phosphoglucose mutase will flip it to the 6th position, and for that it needs to maintain. You need to maintain the anomeric configuration. So that's also pretty cool because phosphorylases does that. But here is some what the phosphorylase will do. It starts clipping from the ends, and now once it reaches, let's say, some branching point, then it moves. Then another enzyme comes in, glucan transferase, and it moves this 3. So all but one of the last ones on the stem there, and moves them to another chain. And then phosphorylase can come in and start clipping from here. But the question is, what happens with the actual little stump that's left behind, the one glucose molecule that is bound to the rest of the chain in a 1,6 glycosidic bond? So that will be cleaved by the debranching enzyme. Okay, but the debranching, and so we have a phosphorylases by phosphorylase removes from the end until it reaches a point where you're really close to a branch, and then a new enzyme, glucan transferase, will come in and move all these 3 residues except the last one, okay, off the branch to another chain. So you have a longer chain now. And of course, this can be acted upon by phosphorylase. And then another enzymatic activity, the debranching enzyme, comes in and removes that little 1,6 glycosidic bond from the stump. It turns out that the glucan transferase and the debranching enzyme, in many species, in most species, are actually separate enzymes, but in animals, it's one. So it's the only monomeric enzyme, one polypeptide that has actually 2 distinct active sites. So there was some gene fusion event at some point in evolution and in the animal lineage, and we have both of them in one protein. So the transferase, it's fairly straightforward. This is the 1,4 bond. It gets clipped, and the last and then another. Sorry, it gets clipped. It's left like this. So this is free here, and then the other glucose from the 4 position from the chain comes in and attacks the 1 position, and now you rejoined it to some other pre-existing chain. Okay. The debranching is nothing but a straight up hydrolysis. So it's a hydrolase. So we don't have phosphorylases at the branches, we have hydrolysis. And as a result, what we have is free glucose. So the one glucose that's bound in the 1,6, it makes a 1,6 glycosidic bond at the branches. When that gets released and mobilized from glycogen, it is released as free glucose. So then you have to use it in hexokinase and phosphorylate at the 6th position and so on and so forth, spend that extra ATP there. All right. So how much of that is in the total? Actually, it's quite a bit. It's about 10%. It turns out all the glucoses in glycogen are branches. So when you clip those, okay, those are released as free glucose. The rest 90%, phosphorylase works on them and you release them as glucose 1-phosphate. And as I told you already, once you have glucose 1-phosphate, you run the reaction as we have seen with phosphoglucose mutase. We're not going to say it again. It's just the intermediate is doubly phosphorylated because the enzyme provides another phosphate on the 6th position, and then the enzyme picks up the original one from the 1 position, and you end up with glucose 6-phosphate, and you're ready to go into glycolysis. And again, the enzyme only regularizes the alpha anomers when the hydroxyl group on the C1 carbon is pointing down. And so it was good that the phosphorylases kept the anomeric form of carbon in the same configuration. So you don't have to deal with that. So we'll close with this, with glycogen degradation. And finally, we're going to see some pretty cool stuff, how the whole thing is regulated in the next video.